collectively at the end of these talks. So thank you so much, Michal. Next speaker is Dr. Jessica Fetterman from Boston University School of Medicine. The title of her talk is Gaining Insights into Mitochondrial Diseases Using Stem Cells.
Thank you so much. I just want to start by thanking Sophia and Casey for giving my research team the opportunity to co-host today's symposium and for the opportunity to present some of our latest research in our lab. So just to remind everyone, Vamsi already did a great job of orienting us, but there are many different organelles within the cells of our body, and there are two that are really important for my presentation today. The first is the nucleus, which is depicted in the middle in purple, and the nucleus contains a lot of the genetic information in our cells. And the other organelle of interest in my talk today are the mitochondria, as is the interest of most of us on this call today. And the mitochondria also contain genetic information in the form of the mitochondrial genome. And so just very simplistically, mitochondria take the food that we eat and convert it into a form that our cells can use called ATP. This process, much like our cars, is not completely efficient. And so some of the energy is lost in this process in the form of thermal energy or heat. And so the way that mitochondria convert the food we eat into ATP involves a process called oxidative phosphorylation. Depicted here in the middle are the oxidative phosphorylation complexes. There are 5 oxidative phosphorylation complexes, and each of these are important in catalyzing reactions that result in ATP production. Uniquely, the oxidative phosphorylation complexes are encoded by 2 genomes. They're encoded by both the mitochondrial genome as well as the nuclear genome. And genetic variants in either of these 2 genomes results in mitochondrial diseases, including Leigh syndrome. And there have been a number of different barriers to advancing our understanding of mitochondrial diseases and mitochondrial genetics in general. The rarity of carriers of very specific genetic variants has made it challenging to identify drug targets and to carry out adequately sized clinical trials. As was mentioned earlier, there are currently no approved drugs for the treatment of mitochondrial diseases. And in part, it's been particularly challenging as the therapeutic responses often differ across patients. And that's likely a result of the heterogeneity in the genetic variants that cause these diseases, as well as differences in the biochemical defects that are introduced by each of these different types of mutation or variants. In addition, the tissues that are most affected in mitochondrial diseases, especially the heart and the brain, are clinically inaccessible. So we can't easily or safely biopsy the brain or the heart of a patient to gain insights into the disease. And so we're really limited to cells that are more peripheral, such as the blood cells. A number of models have been used to study mitochondrial genetics and diseases. Some of them include animal models such as mice, and these are really great for studying mitochondrial genes that are phylogenetically or that are conserved throughout evolution. However, the mitochondrial genome is not very well conserved, and because of interspecies differences, it makes it really challenging to use animal models to study human mitochondrial genetic defects. Another model that's been used are the cybrids. And cybrids allow us to study mitochondrial genetics in a dish. The way this works is you take a cancer cell line, such as an osteosarcoma cell line, and you deplete it of its mitochondria through either chemical or genetic manipulation. The cell with very few, if any, mitochondria left is called a row 0 cell that you then fuse with platelets from a patient. And platelets have no nucleus, so they have no nuclear genome. And so the only genetic material contributed by the platelet is the mitochondrial DNA. And so this ultimately leads to the formation of a cell that has the nuclear genome of the osteosarcoma cell line from which it originated, but the mitochondrial genetics of the platelet. And the use of cybrids has really helped us understand a lot of the basics of mitochondrial genetics, in particular heteroplasmy. But cancer cells are notorious for having metabolic and mitochondrial dysfunction that in part makes them a cancer cell. And so it's really difficult to understand how a particular mitochondrial genetic variant would alter the cell types that are most relevant to the diseases that affect patients, predominantly things like the neurons and the cardiomyocytes or heart muscle cells. And so it's really time that we start to use new models to understand mitochondrial genetics and diseases. And so one of the models that our lab is using are called induced pluripotent stem cells. So induced pluripotent stem cells are derived from either a skin biopsy or from blood cells collected in a routine blood sample collection. Importantly, induced pluripotent stem cells do not require the use of embryos. So unlike embryonic stem cells, there's no ethical controversy in utilizing these cells. And of benefit to the scientific community, these cells, once you create them from a patient sample, they're indefinitely self-renewing. So we can maintain them long into the future without having to go back to the patient and recreate them. And the thing that's really important about these cells is they have the ability to become any cell type in the body under the right cell culture conditions. And so we can actually take induced pluripotent stem cells from a patient and create the cell type that makes up the tissue that's affected in that patient to better understand the disease. And so my lab has been working to take induced pluripotent stem cells and generate cardiomyocytes, or the heart muscle cells. And so pictured on your left is an immunofluorescence image of a cluster of cells in culture after having undergone directed differentiation of the stem cells to cardiomyocytes. And what we've done is we've stained the cells for cardiac troponin in green and NKX 2.5 in red and the nucleus in blue. And what you'll notice is that there are clusters of cells that are both green and red. So they are positive for both cardiac troponin and NKX 2.5. And these are two specific markers of cardiomyocytes and are telling us that we are successfully creating cardiomyocytes from the stem cells. And the really cool thing about these cells is, much like in the human heart, they actually beat in the dish in culture. And I have a video to show this. And so what you can see is in the bottom, you see the start of the contraction that then is transmitted all the way across the well of the plate. And so this really allows us to understand how genetic perturbations affect the function of these cardiomyocytes, which is reflective of what's going on in the hearts of the patients. And so we plan to use induced pluripotent stem cells to really understand mitochondrial genetics and mitochondrial diseases. And so we can do this by collecting a blood sample from patients with a mitochondrial disease. We isolate some of the cells from that blood sample, and then we reprogram those blood cells using 4 transcription factors known as the Yamanaka factors. And that generates our induced pluripotent stem cells, or iPSCs. And these cells carry the genetics of the patient themselves. And so what we can then do is we can use gene editing techniques like the 2 prior speakers discussed in order to correct the pathogenic variant, allowing us to study side by side cells from the patient that have the unedited cells that have the pathogenic variant, and the cells that the only difference is the edited correction of the pathogenic variant. We then take these cells, both the edited and unedited cells, and undergo directed differentiation to cardiomyocytes, much like what I showed you on the previous slide. The next step is deep functional characterization of these cardiomyocytes. And we can measure things like contractility and beat rate to understand cardiomyocyte function. We can look at measures of mitochondrial function, such as mitochondrial respirometry, and perform multi-omics to really deeply understand what are the abnormalities that are occurring because of the pathogenic variant. We can then take this information and identify drug screening strategies. So what we can do is we can say, okay, these are the abnormalities that we've identified. How can we target them with either existing FDA-approved drugs for other conditions or using novel and emerging drug therapies, and screen them in the patient's cells to identify the most effective therapy that would work to treat the patient from which the cells were ultimately derived. And this is really getting at creating a personalized medicine approach for every patient, where you can identify what are the specific alterations that are occurring in the cells from the patient, and how can we best target those to improve the outcomes in patients with mitochondrial disease. And if you're interested in learning more about the stem cells and how we're using them in our research program, we invite you to join us. We'll be hosting a clinic day at Boston University in the spring of 2023, where you can come see our state-of-the-art facilities for stem cell research and learn more about our research program and how you can be involved and participate in our studies. And with that, I'd just like to thank my research team as well as my many collaborators. In particular, I would like to thank Dr. Deepa Gopal, who's my MD counterpart leading our research team, as well as Jesse Morera, who is a postdoctoral fellow who created those beautiful images in the video and has really refined our protocol for directed differentiation of stem cells to cardiomyocytes. And I'd like to thank both Jesse and Karen for doing a lot of behind the scenes work today to help make it go smoothly. And with that, I will stop sharing. Thank you.